As they walked out of Africa, the population that would evolve into both Neanderthals and Denisovans had no way of knowing that the genetic diversity in both their lineage and that of their offspring species would forever nosedive. This is due to many factors. Some are related to the lifestyle of Neanderthals, while others are the result of the nature of population genetics. Welcome to Dawn of Sapiens. In this video, I'll explore the genetics of Neanderthals. Because genetics is intertwined with all aspects of a species, from life history, culture, demography, ecology, to extinction, not every aspect of Neanderthal DNA will be explored in this video. In the first video, I touched on the loss of genetic diversity as Neanderthal populations collapsed and re-expanded over time, leaving only a fraction of diversity behind. What I didn't really touch on is the population bottleneck that occurred when the ancestral population of both Neanderthals and Denisovans trekked out of Africa. In this case, this event can be described as a founder effect. Founder effects occur when a founding population splits from a larger population and remains genetically isolated for an extended period of time. This founding population only takes a fraction of the diversity found in their mother population. Even if this new population goes on to expand to 1,000 or even 1,000,000 individuals, it will have a low diversity for an extended period of time. To put this into perspective, DNA analysis of modern humans reflects an effective population size of 10 to 25,000 people. The effective population essentially consists of reproducing individuals. Generally, this is about 1/3 to 1/2 of the overall population. In other words, children and older individuals are excluded in this figure. When they are accounted for, this increases the overall population size to 30 to 75,000 individuals. Think about that. A population of 7 billion modern humans possess the genetic diversity of at most 75,000 ancient humans. This illustrates just how long it takes for nature to generate genetic novelty. This is a good point to define genetic diversity. Most of the 20,000 genes in the human genome are identical in all humans, but some genes have multiple versions. That's to say, in one gene at a particular location, there might exist a C or cytosine, but another individual might have a different version of that gene in that same spot. These are two different versions or flavors of the same gene. This change may be detrimental, neutral, or beneficial to an individual's survival, and in most situations, natural selection will weed out the detrimental mutations while leaving neutral and beneficial changes in a population's gene pool. In a population with less genetic diversity, individuals will have less flavors of these genes and will have a genome that is more identical. This can make a population more susceptible to disease and less able to respond to a changing environment. Neanderthals, for reasons not yet fully understood, lived in smaller, more isolated populations than modern humans and even Denisovans. Individuals from 5 sites demonstrate the size of Neanderthal populations. Over in the Russian tundra, Denisova 5's effective population is thought to have been between 30 and 55 individuals. But evolutionary geneticist Fabrizio Maffesoni and his team used models to show it was probably closer to 30 individuals. Staying in the Altai region, but moving 65,000 years later, evolutionary geneticist Laurent Scove and colleagues found that the Chagraskaya population was even smaller at about 20 individuals. A rock shelter in Krapina, Croatia, preserved fossils that are estimated to have originated from between 75 and 82 Neanderthal individuals. There's an asterisk on this site because of a strange fact about these remains. In 1979, anthropologist Milford Wolpoff noted that, quote, the age distribution of the sample is unusual in the absence of individuals under 3 and the virtual absence of individuals over 20. The highest observed mortality was between the ages of 15 and 18, end quote. These remains don't appear to be a natural community. Most of them were children up to 18 years old. Was this a cemetery of some kind? The dating of these remains shows a possible range of 10,000 years. Might this be 10,000 years worth of Neanderthal burials? I think in any scenario, this graveyard indicates the presence of a large continuous population for at least several thousand years. Elsewhere in Croatia, geneticist Leming Lee and coworkers show that the 50,000-year-old Vindiha effective population came in at a whopping 62 individuals. In southeast France, the size of Thorn's population is not known, but paleoanthropologist Ludovic Slimak and his team suggest, quote, small group sizes. The available Thorin DNA shows fewer long runs of homozygosity than Denisova 5, but more than VI 33.19. Using this data, I'm going to give it my best guess and say Thorin's population size was between these two populations at about 55 individuals. Literally days before I sat down to produce this video, researchers Liming Lee, Troy Comey, Rob Bierman, and Joshua Aki released an earth-shaking study. It produced several revelations, but there's one that is particularly relevant here. It revolves around the fact that gene flow into Neanderthals from ancient Homo sapiens has mostly been ignored. Because of this, portions of the Neanderthal genome that were previously assumed to be of Neanderthal origin are actually from ancient Homo sapiens. This means that all the previous studies on Neanderthal DNA were overestimating the genetic diversity of Neanderthals. Lee's analysis indicates that the size of the overall Neanderthal effective population is a staggering 20% smaller than all previous predictions. That means the sizes of these five populations are likely even smaller than I've just demonstrated. These five populations are the easiest to illustrate typical group sizes of the species. But every analysis of both high coverage Neanderthal genomes and low coverage fragments show that low genetic diversity was always the reality of Homo Neanderthalensis. Mafasoni et al. Put it this way: Neanderthals experienced a low effective population size for the last 200,000 years, including the time shortly before each individual, VI 33.19, Chagraskaya 8, and Denisova 5, lived. End quote. Small, isolated groups result in less genetic diversity, which makes individuals susceptible to disease. But the Neanderthals' habit of living in small, isolated groups impacts the species in an arguably even more harmful way: genetic drift. The basic definition of genetic drift is the change in frequency of an existing gene variant or allele in the population due to random chance. This randomness can be caused by multiple sources. For example, random deaths due to freak events can cause genetic drift in small populations. But an even greater source of genetic drift is meiosis, or the process where a single cell divides twice to produce four cells containing half the original amount of genetic information and ultimately produce sex cells or gametes. These sex cells contain only half the number of chromosomes of other cell types, since the second half will come from the other parent. Each parent themselves have two alleles for every gene, which was inherited from their own parents, and which of these two alleles happens to be passed down to the sex cell is said to be random. Bear with me, there's a point to this quick biological digression. When a heterogeneous individual, or someone that has two different versions of the same gene, passes down their genes, their child only inherits one of the parent's alleles, since the other allele is inherited from the other parent. Let's say a heterogeneous parent's genome contains allele A and allele B of a gene, but they only pass down allele B. This means allele A goes extinct in this familial lineage, unless the child happens to inherit it from the other parent. During meiosis, the randomness of inheritance precedes the force of natural selection. Natural selection can only work with alleles that a new organism inherited from mom and dad. The allele that a parent passes down is random and may be beneficial, neutral, or even detrimental to survival. Here's where small populations become important. In a large population, if parents fail to pass down one or more alleles to their children, those alleles become extinct in the immediate family. But there is also a high likelihood that those alleles persist in the overall gene pool of the population. In small populations, not only is there a higher chance that those alleles disappear in the familial lineage, but also in the population as a whole. And this randomness means alleles that do survive in the gene pool may actually be detrimental. In small populations, genetic drift is not fully bound by natural selection. The randomness of inheritance means in these groups, quote, bad alleles can become fixated at 100%, and healthy alleles can disappear altogether. We've already seen that Neanderthals lived in small populations. Despite its role mostly being overlooked in pop science, genetic drift is nearly as strong a player as natural selection in the evolution of Homo Neanderthalensis. Evolutionary geneticist Sergi Castellano and his team argue that genetic drift degraded the quality of Neanderthal genomes. The influence that genetic drift plays in evolution is mostly overlooked in favor of the sexier natural selection. It's much the same in the story of Neanderthal evolution. One inevitable effect of living in small groups is the reality of inbreeding. Neanderthal populations have varying degrees of inbreeding, but overall, these levels are far greater than found in both ancient and modern Homo sapiens. For example, in mitochondrial DNA, Neanderthals differ from each other by 20 bases, while modern non African Homo sapiens differ from each other by 38 bases, and modern Africans differ from each other by 76 base pairs. On an individual level, inbreeding is the quickest way to reduce genetic variation in the next generation. Closely related individuals will have DNA that has many of the same gene versions and therefore less variation. Researchers refer to these identical segments that both parents pass to their children as runs of homozygosity. The more runs, the more inbreeding has occurred. Long runs indicate alarming levels of reduced variation and recent inbreeding. For example, Slimak says this about Thorin, quote, Thorin harbors about 7% of its genome in homozygous segments of 5 megabases or longer, including 45 megabases in segments longer than 20 megabases, indicative of recent inbreeding. End quote. In the end, inbreeding both reduces variation and increases deleterious alleles in Neanderthal groups. The ancestors of Neanderthals had no way of knowing the challenges their DNA would face in Eurasian environments. The founder effect immediately reduced genetic variation when Neanderthals' foremothers and forefathers split from a mother population. At least 3 population collapses further bottlenecked Neanderthals. Each time this happened, even more genetic uniqueness was lost. Having a small overall population paired with very small isolated subpopulations allowed genetic drift to overtake natural selection at times and bring deleterious alleles to fixation in Neanderthal genomes. And finally, inbreeding slammed in the Neanderthal gene pool by further collapse in variation and introducing even more detrimental alleles. It's one thing to talk about the genetics of Neanderthals from a bird's eye view, but the view from that perch makes it difficult to relate to these effects on a personal and individual level. Luckily for us, researchers have teased out examples from the DNA of Neanderthals throughout Eurasia. A gene creatively named sodium voltage gated channel alpha subunit 9A, or SCN9A, manifests in 3 different versions that are distinct from those found in Homo sapiens. But there is a small caveat that some modern humans inherited these alleles from Neanderthals. What's amazing is these Neanderthal alleles result in the nerves connected to these particular pain receptors being highly excitable. This means all Neanderthals likely felt more pain than we would when exposed to the same exact stimulus. The knee jerk reaction to this fact would be to assume that natural selection drove these three painful Neanderthal alleles. It would be a reasonable hypothesis. On the other hand, we've already seen that genetic drift is responsible for a large portion of the Neanderthal genome. This would be an extremely unlucky and unfortunate result of genetic drift's inherent randomness. To add another dimension, the modern human versions of this gene are the same versions found in all monkeys and apes. These sapiens variants appear to be highly conserved across all apes and monkeys, and yet somehow Neanderthals diverged completely from the ancestral variants. Was their environment so different than that of monkeys, apes, and modern humans that increased pain sensitivity afforded them an adaptive advantage? Or did the small, isolated populations of Neanderthals allow genetic drift to drive these alleles to fixation? I have no answers, but these mysterious differences are quite profound nonetheless. They also speak to a difference of experience when compared to us sapiens. If we zoom into the Mesmaskaya site at around 44,000 years ago, the DNA of Mesmaskaya 2 shows an interesting mutation. In a previous video on Homo erectus, I discussed a protein, AMELY, that is found in tooth enamel and only produced by a gene on the male Y chromosome. The presence of this protein indicates a male, while the absence indicates a female. Mesmaskaya 2 upends this approach since researchers observed an AMELY deletion on his Y chromosome. The AMELY gene is essentially missing from Mesmaskaya 2. The DNA of Mesmaskaya 2 confirms he was male, but the enamel protein indicates a female. In the absence of DNA, researchers would wrongly conclude this individual was a female. This is a great example of why multiple lines of evidence are so beneficial to scientific research. In 2023, Claire Demarche and colleagues released a study on the genetics of odorant receptors in Neanderthals, Denisovans, and modern humans. They not only compared the sequences of 30 odor receptor genes in all three species, but they functionally tested 10 variants from 8 genes. They found that all three species were capable of detecting the same odors, but at different intensities or sensitivities. Compared to modern humans, Neanderthals had an especially blunted response to spicy, mint, herbal, and spearmint smells. This is even more true with balsamic or vinegary odors. The authors show that for balsamic odors, modern humans score a 26.41 on what they call the activity index, while Neanderthals score a mere 2.0. There's not enough research for me to make an argument one way or the other, but I can't help but feel that a lower genetic diversity played a part in the sensitivity of Neanderthals' sense of smell. Overall, all measured odors scored lower in Neanderthals than in modern humans. Was it not for the fact that the Denisovan to modern human comparison is much more variable, one could wonder if us moderns evolved a higher smell sensitivity rather than Neanderthals possessing a duller sense of smell. But it just doesn't seem to be the case. In every comparison, Neanderthals scored less on the activity index. Regardless of the driver of this difference, you have to wonder how it affected their experience of the world around them. Did it decrease their hunting efficiency? Anyone who has had a stuffed nose knows that food tastes completely different. Did this difference in smell intensity mean foods tasted differently to Neanderthals? As one of the proverbial five senses, it's easy to argue that their lack of sensitivity was a net negative. This study is another reminder that while we are rediscovering Neanderthals to be more similar to us modern humans than we could have ever imagined, the way they interfaced with the world was also uniquely their own. In 2020, evolutionary anthropologist and geneticist Hugo Zeeberg, Jeanette Kelso, and Nobel laureate Svante Pabo released a study on the progesterone receptor of Neanderthals. They found that the Neanderthals carry a unique allele of the progesterone receptor gene known as V660L. The Denisovan 5 genome shows this allele was homozygous. She inherited the same version from both parents. The other two high coverage Neanderthal genomes, Chagrskaya 8 and Vindiha 33.19, also show this allele being homozygous in their genomes. While SD1253 hasn't produced a high coverage genome, enough of it was captured to show that it also inherited this allele from both parents. The genomic fragments of Mesmaskaya 1, Mesmaskaya 2, Q56-1, Z4-1514, and Denisova 11 can only show that they all inherited this allele from at least one parent. Yet, at every possible opportunity, DNA analysis only finds this allele in Neanderthal genomes. This is a strong indication that this allele is fixed in Neanderthals. To put it into perspective, the only high coverage Denisovan genome does not include this Neanderthal variant. Naturally, the next question becomes, what does this gene and this allele in particular do? We don't have Neanderthals to participate in clinical studies or Joe Rogan's favorite double-blind placebo-controlled studies. Fortunately for us, some modern humans inherited this allele derived from Neanderthals. In modern humans, the Neanderthal version results in less hemorrhaging in early pregnancy and in less miscarriages overall. Surprisingly, Zeeberg and coworkers found that modern humans with this Neanderthal allele have more sisters than modern humans with the modern human variant. This combined effect of a reduction in miscarriages and increased females being born seems to improve the overall fertility of the Neanderthal species. Once again, one must ask, is this a product of genetic drift or natural selection? Just like the SCN9A pain allele, V660L appears to have become fixated at 100%. On the surface, this allele seems to increase fitness of the species rather than degrade it. Did the randomness of genetic drift do the Neanderthals a favor in this instance? Or is this increased fertility a result of natural selection? Obviously, I can't know for sure, but it's hard for me to conclude that this was a result of genetic drift. I'd even go as far as saying this adaptation probably proliferated because it was able to offset the genetic issues in the Neanderthal gene pool. Low genetic diversity, inbreeding, malformations, and other issues are ever present, but the more surviving individuals, the more opportunities there are for beneficial mutations to bubble up from the void. I can't help but feel that this mutation arrived randomly but quickly spread in proto-Neanderthals as it helped to counteract the vulnerabilities of the species gene pool due to genetic drift and low diversity. In the early 1990s, scientists became interested in a British family they dubbed the KE family. Multiple generations of this family appeared incapable of talking. These individuals displayed impaired cognitive processing and orofacial dyspraxia, or a lack of control of the movement of their jaw, lips, tongue, and soft palate. To make a long story short, researchers pinpointed the issue to a mutation in the FOXP2 gene. It's for this very reason that the gene became known as the quote, language gene. A bad version of this gene renders individuals unable to communicate. Some imaginative thinkers took this notion of a language gene to the extreme. It became popular to conjecture that once this quote, human version appeared, our ancestors magically transformed from grunting cavemen to swooning troubadours. Of course, this led to everyone wondering, did Neanderthals carry our version of this gene? The proteins that this gene produces have 3 amino acid differences between human and mouse versions. Human and chimps differ at only two amino acids. Neanderthals and humans have the same protein, as do Denisovans, but there is some thought that a difference does exist in the regulatory portion of the gene. It has yet to be fully identified. The thing about genes is they can be used or expressed in multiple tissue types. This hampers their ability to mutate because while a mutation might amplify a function in one type of tissue, it might totally destroy a function in another tissue where it is used. It also complicates natural selection. If researchers detect a strong signal indicating increased frequency of a gene variant, they then need to determine which function and in which tissue a selective pressure is acting on. No longer can you argue the effects of one gene mutation on one behavior or phenotype. A mutation could affect several tissues and behaviors at different intensities. Although FOXP2 is a protein, it functions very different than you might imagine. When we think of proteins, we often think of the physical structures of the body. Skin, muscle, and bone are all largely protein derived. But FOXP2 has more in common with a hormone than it has with the muscle. The protein that the FOXP2 gene produces functions as a transcription factor or a chemical signaler that turns other genes off and on. It plays a major role in the development of the brain during pregnancy. In fact, the expression and ensuing results of brain tissue has been shown to be very similar in humans, monkeys, rats, mice, zebrafishes, and birds. The portions of the brain that FOXP2 is responsible for forming in all these species are the same portions of the brain that show abnormalities in the KE family. That's not to say there isn't any difference between the human version and the versions of other species. The human version expresses 116 neuronal genes differently when compared to the chimpanzee version. The FOXP2 differences between chimps and Homo sapiens is expressed as far as the FOXP2 protein. I've already said the FOXP2 protein of Homo sapiens and Neanderthals are identical. This is true, but there appears to be a single letter difference in the Neanderthal FOXP2 allele and the allele of Homo sapiens. This mutation sits at position 114,076,877 in intron 8 of FOXP2. The primitive or ancestral allele has an adenine at this position, and the derived or modern version contains a thymine in this spot. This stretch of DNA is actually where a receptor or binding site sits for another transcription factor called POU3F2. We might as well think of this spot as one of probably many dials that rev up or turn down the FOXP2 gene. Interestingly, our thymine version renders this quote dial 40% less effective as the ancestral allele. In other words, the POU3F2 signaler has less effect on the protein output of our FOXP2 gene. According to Maricic et al., POU3F2 only binds to the FOXP2 gene in neurons, and the ancestral version existed as the sole version for 700 million years until humans wandered into the picture. The pop science story goes that this allele appeared between 150,000 years ago and swept through the population. This eventually gave us language and superior communication compared to other hominins. This narrative wasn't pulled from thin air, as Maricic et al. And others detected a selective sweep between 200,000 and 50,000 years ago. However, in 2018, Elizabeth Atkinson and her team re-examined the genetic history of the FOXP2 gene with modern genomic technology and larger sample sizes. They found that the small sample sizes, something like 20 individuals, and their predominantly European ancestry produced a statistical artifact that resulted in a false signal of a recent selective sweep. When they reanalyzed the gene with a sample size in the thousands and from individuals spanning multiple continents, they found no sign of a selective sweep. In fact, they discovered the quote modern human allele knocking about in the genomes of several Neanderthals and Denisovans, including one copy in the Alti Neanderthal. Realizing that some Neanderthals and Denisovans carried the quote modern FOXP2 allele makes it tempting to ask, were some Neanderthals able to talk while others weren't? Were there enlightened individuals walking amongst a mass of dullards? Well, without living Neanderthals and Denisovans to experiment with, we'll never know for sure. But what if some existing humans today possess the quote Neanderthal version, or more accurately, the ancestral allele? As it turns out, there are a substantial number of individuals with the ancestral allele, and none of these people have issues operating their tongues, lips, or soft palates and speak just fine. The ancestral allele results in an increase of the FOXP2 protein, while the mutation of the KE family results in a loss of FOXP2 protein because one or both alleles are inactive during their brain development. Living humans and Neanderthals produce identical FOXP2 proteins. The difference in gene alleles resulted in a difference of protein levels during brain development. But the significance of this point becomes blurred when you consider the fact that some Neanderthals carried the quote modern human allele and some modern humans carried the quote Neanderthal allele. And these humans are none the worse for it. Add in the realization that the FOXP2 gene is highly conserved across 700 million years and that the supposed recent selective sweep was an illusion, and all of a sudden, any differences of FOXP2 between Neanderthals and Homo sapiens lose almost all relevance when it comes to language. If speaking is the act of vibrating and encoding air with information, then hearing is the act of deciphering this vibrating air. But the Mesmaskaya population hints that this second aspect of verbal communication may have been a struggle for some Neanderthals. Evolutionary geneticist Tatiana Andreeva and her crew found that the early Mesmaskaya population inherited a mutation that degrades the ability to hear. This mutation is very rare in modern humans, but those that do possess it can experience no symptoms at all, complete deafness from birth, mild deafness, or late onset deafness. In the mitochondrial DNA, specifically at position 1555, the ancestral adenine base mutated to a cytosine. And this mutation has a curious relationship with compounds known as aminoglycosides. For instance, even with perfect hearing, deafness can be induced in a carrier of this mutation if they come in contact with aminoglycosides. When a 12-year-old Argentinian girl who happened to carry this allele injured her hand, it was injected with antibiotics that contained these compounds. 30 days later, she began experiencing hearing loss in both ears. When this scenario played out with multiple individuals, the hearing loss was observed to be progressive in all of them. Some individuals with this mutation had various degrees of hearing loss even without encountering aminoglycosides. But most individuals with this mutation who never contact these compounds have normal hearing. Did Neanderthals use these specific antibiotics? Without a pharmacy in sight, probably not. However, Andreeva et al. Speculates that if carriers of this allele ate mushrooms containing aminoglycosides, they could unwittingly be initiating their own hearing loss. This mutation by itself increases the likelihood of complete deafness or hearing impairment to various degrees. If certain mushrooms were eaten, the chances increase. The fact that this mutation actually sits in the mitochondrial DNA increases its potency. How is that? Because mitochondrial DNA is passed down as a direct copy from mother to offspring. Unlike the X chromosome and autosomes, mitochondrial DNA does not undergo recombination or meiosis, and the child doesn't receive a copy from each parent. The child has to live with the mitochondrial DNA that mom passes down. When I realized this mutation existed in the mitochondrial DNA, I found it fascinating because it meant not just MES3, but also her mother's entire lineage were likely carriers of this allele. When I tried to confirm this in Andreeva's paper, I was surprised that it was barely discussed. I was even more surprised to find one of the few descriptions characterizing the 827G 12S rRNA variant as being, quote, fixed in Neanderthals. Could it be that all Neanderthals were much more likely to suffer from impaired hearing than both Homo sapiens and Denisovans? I found very little when I tried to locate more research on this gene and the Neanderthals who carried it. I was tempted to email Andriva and ask, or to just leave it at that and assume that the whole species had an issue with impaired hearing. But then I remembered a graduate course I took at Arizona State University called Functional Genomics. Because most DNA databases are publicly available, I decided to try and figure it out myself. On the National Library of Medicine's nucleotide viewer, I pulled up the reference Neanderthal mitochondrial DNA. At position 1555, this genome showed the mutated version. But knowing that the Homo sapiens reference genome is actually a composite of more than 8 individuals, I didn't trust this Neanderthal reference as showing the full picture. If the Neanderthal reference genome is a composite of multiple individuals, you can never be sure which allele from the multiple individuals they chose to represent for each position in the reference genome. After a little digging, I found this is, in fact, the case. Instead, I analyzed the mitochondrial DNA of several Neanderthal individuals belonging to waves 1 through 4, as I described in the previous Neanderthal video, along with one individual that wasn't featured in that video. The AT6672 population was the only representative of the Proto Neanderthals available. Of the early Neanderthals, I found the mitochondrial DNA of SCI-4A and S5000. I was actually surprised the database didn't seem to have the mitochondrial DNA of Denisova 5, whose genome is sequenced at high coverage. But I did find the mitochondrial DNA sequence for Denisova 17. Denisova 17 is a Neanderthal individual whose DNA was sequenced from long bones. It was distantly related to Denisova 5 and lived in the same cave 134,000 years ago, or 14,000 years before Denisova 5. The mitochondrial DNA of MES3, of course, was in the database. MES3 is interesting because I have it classified as a classic Neanderthal, but depending on how you look at it, her lineage is a bridge between early Neanderthals and classic Neanderthals, and could be classified in either group. The rest of the classic Neanderthals, which I found mitochondrial DNA for, are Chagroskaya 8, Okladnikov 2, and Vindiha 33.19. The terminal Neanderthals are represented by Q56-1, Z4-1514, Vindiha 33.25, and SPY 94A. I found that AT-6672, Denisova 17, SCI-4A, and S5000, all of which lived between 430 to 116,000 years ago, had the ancestral adenine at this position. These populations were not inherently vulnerable to increased hearing impairment or loss. Of the classic and terminal Neanderthals, MES3, Chagroskaya 8, Vindiha 33.19, MES2, Q56-1, Z4-1514, and SPY94A all possess the mutated allele with the cytosine base at this location. These individuals lived from 100,000 to the last days of the species. When taken together, an interesting and familiar pattern emerges. The first set are members of what I've classified as the Proto and early Neanderthals. As I discussed in part 1, these two groups possessed much more genetic diversity than later populations. By 110,000 years ago, these early Neanderthals had disappeared, and a group or close network of populations expanded to repopulate much of Eurasia. Mesmeskaya 3 is dated to about this time, and represents the first member to show this mutation. It seems that when Eurasia was repopulated, this mutation was fixed in the expanding groups. That's fascinating, A large fraction of Wave 3 and Wave 4 Neanderthals likely lived with various levels of hearing impairment or deafness. Even speaking that sentence instinctually feels like hyperbole. Yet, this mutation is found throughout the genomes of many Neanderthals, and the natural experiment in modern humans does demonstrate the unfortunate effects of this mutation. Remember the AMELY deletion I discussed earlier? It actually has a part to play here, or at least I thought it did. As I was researching that mutation, I was surprised to see researchers asserting that its deletion increases the chance of late onset hearing loss in modern males. The absence of MLY isn't directly responsible for this hearing impairment. Instead, a gene called TBL1Y is the main influence. Like MLY, the Y in TBL1Y indicates it sits on the Y chromosome. Jobling et al. Found that in modern humans, anywhere there's an MLY deletion, there is also a TBL1Y deletion. This is because TBL1Y literally sits right next to the MLY gene on the Y chromosome. Bassi et al. Revealed the deletion of TBL1Y is associated with progressive hearing loss in males beginning in their 30s and 40s. I found this possibility of Neanderthals having another mutation that induces deafness almost unbelievable. And when I returned to the original study that sequenced the Mesmoskai 2 genome, it turns out their MLY deletion is slightly different than that found in modern humans. It just so happens that the MLY segment does go away, but in Neanderthals, the deletion ends before the TBL1Y segment. Luckily for Mesmoskai 2, he escaped further potential hearing impairment as he was spared the disappearance of his TBL1Y gene on his Y chromosome. But the fact remains that all of these classical and terminal Neanderthals were burdened with hearing problems thanks to the mitochondrial mutation. Might this mutation be a prime example of both bottlenecks and genetic drift? This could be true, but it also might be true that the mutated mitochondrial allele didn't completely fixate at 100%, because the 53,000-year-old Okladnikov 2 individual, along with the very late 40 to 35,000-year-old Vindia 33.25, both possess the unmutated allele. More importantly, what did the pervasiveness of this condition mean to the life of classic and terminal Neanderthals? How did they adapt? Was this distinct biology reflected in cultural practices? Perhaps deaf individuals remained in camp as shamans, babysitters, guards, or entertainers. Did impaired hearing generate unique hunting techniques not known to earlier Neanderthals and Homo sapiens? The stories hidden within DNA of Neanderthals tells a story of a species that is both familiar to us sapiens, but also a tale of unfamiliar realities. By stepping out of Africa when they did, the common ancestor of Denisovans and Neanderthals initiated a future that would forever be dictated by the bottleneck from leaving Africa, the resulting low genetic diversity, the so far unexplained small populations, the inbreeding within many of these tiny groups, and the outsized influence of genetic drift. The sequence of adenines, cytosines, guanines, and thymines of the Neanderthal genome hints at the consequences of the bottlenecks, population collapses, inbreeding, and genetic drift that would define the life history of Neanderthals. Some of these consequences are negative, some are neutral, and others are positive. The SCN9A mutations, which result in increased pain sensitivity, likely reach fixation due to genetic drift. Except for Neanderthals, this gene sequence is highly conserved across monkeys and apes. The AMELY deletion can probably be considered neutral, as its absence does not noticeably alter the morphology of the dental enamel of male Neanderthals. In most cases, Neanderthal odorant receptor variants result in slightly blunted smell sensitivity. In several cases, it is extremely blunted. The way they interfaced with the world around them was slightly different. The V660L Neanderthal allele both decreases bleeding in early pregnancy and decreases miscarriages. Because this allele increases fertility, it's hard for me to not consider it as a positive mutation and a counterbalance to the genetic imperfections of Neanderthal genomes. While the Neanderthal FOXP2 variant is distinct from Homo sapiens, its presence in some modern individuals demonstrates it does not inhibit speech and communication. What do the genetic realities of Neanderthal population dynamics and evolutionary histories, along with all these examples, tell us? From my perspective, these realities three dimensionalize, which isn't even a word, but it should be, the life of these Eurasian hominins. Ancient DNA has allowed us to peek into the lives of Neanderthals in ways that would otherwise be impossible. In this video, I barely scratched the surface. Because DNA is forever intertwined with culture, ecology, biology, and population demography, the genetics of Neanderthals will continue to be a prominent player throughout this series. Thanks for watching. If you found this video valuable in any way, please consider liking and subscribing.